Yep, so lucky last, I'm going to not talk about celestial designers, but something a little bit more at ground level and perhaps below ground level, in the sense that I want to be, just to bring you up to date with the state of play of the CellML project, which is really designed as a mechanism that will allow the community, indeed the cardiac physiome community, but a much wider community than that, to exchange models to be able to represent the mathematics and the topology associated with models, so that we can make progress with science and not actually have to worry too much about how things are done. But what I'll do here is talk a little bit about the background of CellML, and then where we are at and what are our current activities. Okay, so we're all aware of the difficulties of the sort of traditional publication process. How do we get our ideas of science from our lab out into the wider community? And the traditional approach, of course, is publish, publish, publish. But that publishing process is indeed very error-prone. There are many steps where we can run into problems. Formulation: the developer is in developing the model, has to write out the equations or determine the equations, and then write up a paper that gets submitted to a journal, whereupon the referees are looking at these equations, and finally down to where we bring it out into the community and people want to use those models. Each of these steps here involves human intervention and interpretation, and the possibility, indeed the likelihood of errors creeping in. So CellML was actually developed around about 10 years ago, at least has been developing over the past 10 years, in order to address this very problem. Right from the outset, we tried to make the language for describing the mathematics of biological models as simple as possible. So there was this idea, we wanted to ensure that whatever we describe for the language to represent these models should be as minimalistic as possible. It really is focused on describing the mathematics and the topology of models. So what does it include? Well, it includes information about the model structure, that is, how we have the parts of a model interacting. It includes the mathematics, because mathematics is the way in which we describe quantitative relationships, and we've done that for many years in publications. There is absolutely no reason why we should reinvent that commonly accepted and understood mechanism. And then finally, metadata, information about the things that we're describing. So that is a very sort of wide area that I'll talk a little bit about later. So CellML includes mathematics and metadata by taking advantage of existing standards. We use Content MathML as our vehicle for describing the mathematics. That's a well-established standard. SBML, for instance, uses exactly the same technique. It's a very sort of coherent way of describing at least a part of the mathematics that is widely used in the publication. We also use existing standards like XLink and more importantly, RDF, Resource Description Framework, for describing the metadata. So, I just want to point out here the simplicity of CellML itself. The basic unit of a CellML model is the component. It has units, variables, mathematics, and metadata. It's basically just a little container for some ideas. And it's got an interface. This is what is available to the outside world. Well, that's fine, but normally models involve several ideas, interacting components, so we have these connections that enable us to link to the interfaces of various components. One thing to note is that all of the variables in the interfaces of the components are required to have units associated with them. We've done this as a deliberate policy to ensure that components made by one person can interact in the correct way with components made by another person, and we allow then automatic units translation. Okay, and we enable hierarchical models to be built by providing two different types of interfaces to components. Public interfaces are what everything can see. The private interface is what you want to hide from the rest of the model. So by having these two types of interfaces, we provide a mechanism for building hierarchies, providing a level of abstraction that you would not otherwise be able to achieve. And then the model is simply a container where we throw all of this stuff in a nice convenient package, typically a file that you can fire off to your colleagues to run or interpret. There's one further level of sort of encapsulation, which is designed to provide a mechanism for reusability, and that is the idea of building supermodels from submodels using something called an import facility. So this might be a model that has been tried and tested by one group, that's packaged in a file. Another model which may be dealing with some other aspect of the physical processes that you're interested in, again, can be tried and tested. But now a new modeler might come along and say, well, I want to make use of both of these concepts. Well, you can use the import facility to tie those together and use connections to bind the relationships between those two models. This is an essential aspect of reusability. Okay, so some of these ideas I've already talked about, but basically I want to emphasize that the CellML itself has a very simple structure that encourages modularity. It encourages the idea, or supports the ideas of decomposition and abstraction and the building of hierarchies. Also, this idea of reusability is supported because we have a publicly available repository of well-tested models in something called the CellML repository, available at this website here. I think this is central to enabling the scientific community to be able to avoid having to duplicate the work that many others have done and give them access to very reliable and robust tested models. Okay, so bringing this slide up here, just a screenshot of one of the models that we've got in the repository. We have just upgraded the repository. It's become available over the past week, and it is a major improvement on the Mark I repository because the new repository here has facilities that allow us to handle these hierarchical models, building models out of other submodels, and also it allows the creation of workspaces so that people, for instance, or groups of people can share workspaces and control accessibility to the models that they are building, and yet take advantage of all of the other models in the repository. Associated with the model as a repository, as I indicated before, is a curation process. And this is central to giving people the confidence of that you require in order to use the models within the repository. So we're going towards Miriam compliance. There's a fairly well-defined minimum information required for models. At the moment, we're indicating the level of confidence or the level of curation by a star system. The other thing I'd like to mention is we've just opened up a new CellML website, which hopefully will give the community a much easier way of finding information about CellML and the models available through the Physiome project. Once again, we're trying to support community efforts by providing special project pages if required, so that the collaborators can be explicitly a part of the CellML website. There are many simulation and authoring packages available. This information is available under in the website under the Tools section. An example of a tool that is available is a collaboration between Auckland and Oxford. This is something called OpenCell, which provides quite a nice integrated environment for authoring and simulating rather complicated models from the repository. This here is some output of a model that has recently been published by Jonna Terkelsen et al. Not everyone wants to use these sorts of environments. You already have, some work tools that you are building or others have built for you. To solve the systems of algebraic ODEs. Well, how do we accommodate that? Well, we've made available also an application programming interface that provides a large number of the tools that are required in order to read in CellML files, process them, and make them available in a form that your application requires. It also has facilities like, for instance, creation of the, say, Fortran or C or MATLAB code directly from the declarative descriptions of the CellML models. We're trying to support community efforts here via a number of mechanisms. As I mentioned earlier, the making available the sites within the CellML website, but also via discussion lists and also a feature tracker. So if you want to request features, then this is the place to go. Okay, so one of the things that is a consequence of having a very minimalistic approach to creation of CellML is the need for adding further semantic information to models. And again, this is facilitated by linking in ontologies to the files. I had an example here of how we are creating our own ontologies and also linking into anatomical and biophysical ontologies like foundational model for anatomy, systems biology ontology, and the ontology for physics and biology. But I don't have time to run through that, so I had better just skip these slides here. Our current work really is centered around trying to make the language simpler, actually, but trying to also accommodate the needs of the modeling community. In particular, we're trying to make the language simpler by removing domain-specific ideas from the language and trying to push those down to linkages supplied by ontologies. However, one of the things that we are cognizant of is the inability of content MathML, for instance, to handle delayed variables, and that's one of the high priority additions that we've got in our current update. So currently also we're trying to provide a strong push to decompose our existing models into reusable small components, like down to the level of ion channels, etc., that modelers can make direct use of. And also there's a strong push to provide the editing and authoring tools and annotation tools available to the modeling community. Many people involved in this. We have around about a dozen workers pretty well full-time, but still we want to have the involvement, further involvement of the wider community in this effort. And then finally some linkages. I thought at this meeting it might be a good idea to have an instantiation of the virtual physiological Newton. Okay, so that's it.
I'm talking as an experimentalist now, because you talked about modelers using this facility, but experimentalists like to be able to go and get the models to get an interaction with their experimental work. So as a skeptical experimentalist, what is the process of gatekeeping? So what's going in is not junk in, junk out. So how do we get confidence that you've got some control over the quality of the model?
Yeah. So part of the motivation for the new repository is to provide the workflows associated with this. The process at the moment is, you submit the model to our reviewers, who will run the model and see whether it corresponds to public standards. Yeah. That's a pretty time-consuming effort, and at the moment relies on relatively small number of people. Once they have the confidence of that the model is producing the right solutions, then it gets a curation tag and placed on the public site. We're hoping to devolve that work down to a larger number of experts within the particular communities, and once again, that requires the input from the wider community.
I would like to follow up also from experimentalist point of view. So we're trying to put together a model, if you wish, of experimental data, which would put together anatomical structure with heterogeneous expression of RNA and protein, as well as function. So example I gave during my talk, the action potential, for example, across the wall is
yes.
distributed in a certain way, but it's only in this particular part of myocardium. And if we isolate cells from another part of myocardium, so we may have some other experimental data, and we would like to accumulate this data specifically from the human heart. We would like to integrate with mathematical models in a certain framework. So do you think in this direction how to integrate actual experimental data of different levels with virtual heart models?
So that is slightly outside of the scope of the CellML effort, but there are other efforts that are trying to address that. In particular, the field ML and biosignal ML efforts are well underway to provide the standardized formats. At the moment, the physiome model repository is really CellML centric, but only because that's there are many models available in CellML. Ultimately, the repository will accommodate the field ML and biosignal ML contributions.
Great. Maybe I think it'd be nice to break for coffee and have questions. So the boss has just said if we can come back at 11:40, please, to start the final session. So I'd like, on your behalf, to thank all the speakers